Hello, my name is Alexis Zapata. I'm with the University of Florida, and I'll be evaluating population estimation methods for southeastern pocket gophers. While the mounds of southeastern pocket gophers make them relatively easy to detect, it's hard to initially capture, mark, and then recapture individuals for research. Pocket gophers can create multiple mounds in a singular burrow, which can specifically make population estimation for this animal difficult. This project will evaluate two methods of estimating populations of pocket gophers separately and discuss the efficacy of each, and then finally recommend a method for future research. Although they are considered pests by some landowners, pocket gophers are seen in conservation as ecologically important for soil development, and they can even form symbiotic relationships within the ecosystem. As an example, their empty burrows can be used by other burrowing animals as shelter. They also can promote species diversity for organisms such as plants by aiding with seed and pollen dispersal. If you look at the bar graph I've included on the tallgrass prairie in Kansas, it depicts the amount of plant biomass relative to the distance from gopher mounds and burrows, which are shown in green and white respectively. This shows that the amount of plant biomass is at its greatest in the immediate area around the burrow and the mound. The first method that I want to discuss involves these specialized live traps that were made specifically for pocket gophers, and they're modified piffle traps, and they consist of a bucket trap with a large curved tube. The curved tube is fitted into the tunnel opening that is formed after you dig down into the mound, and you can see a depiction of a mound and the tunnel you dig into below. The concept behind it involves the gopher going up to the opening to plug the hole that you've made in its tunnel, and then it will hopefully fall into the tube and into the bucket. Originally, the traps were not baited since it was found to be ineffective, but the discussion portion of the paper noted that the bucket trap can be accommodated for food. A suggestion could be made to bait the traps and mark for recapture. Factors that increase overall efficiency of this trap can include live capture with little to no harm done to the pocket gopher since there's no spring mechanism on the trap, easy transport to and from field sites due to lightweight material, and it also allows for insulation and some protection from overheating due to the soil that is placed on top of the trap to disguise it and to help plug the tunnel hole. It allows for easy removal as well of the captured animal, which decreases the stress of the gopher. There is no actual suggested method, however, for recapture or marking with this trap, since it mainly focuses on the capture rates. It also must be checked often, every 2 hours, so that means long hours of trapping and resetting possibly. It also is more expensive than other traps like heart traps. The next method I want to discuss involves fluorescent gel or powder, which can be used to track animal movements and in the case of fossorial animals can show whether burrows are active. You can use varying colors and color combinations to differentiate between the individuals, and in the case of pocket gophers, it can possibly give an estimate on how many individuals in the population are using certain burrows, since multiple can live in a single burrow during certain seasons. This could give a more accurate count in comparison to the old method of getting a census count through just the mounds alone. And you would be able to record which individual used which mound by the fluorescent traces left behind. This method allows for the possibility of not needing to capture the animal. You can just place the gel or powder at the entrance of the burrow. And it's also non-toxic and non-permanent, so it won't cause any lasting stress or inhibit the gopher in his daily life like a collar or tag could. The use of the gel or powder can also track through vegetation and soil, where other methods make it hard to detect movement among vegetation. A drawback is that they must be monitored often and checked for all around the burrow since the gophers can leave tracks from foraging further than just the entrance of the mound, and they might require large amounts of the product to track consistently. New soil might also have to be turned over or brought in to cover up remaining fluorescence from previous samples, and it does not help indicate or account for movement unless a long-term study is performed across seasons. On its own, although the specialized live trap lacks a specific method to mark with the trap alone, it allows for recapture assumptions to be met, such as the possibility of only 2 captures being needed, in which the Lincoln-Peterson method could be used for data analysis. With the powder gel, it lacks a capture component to get a more exact population estimation by itself. But on the other hand, the use of it would allow for less disturbance to the pocket gopher, other than marking methods, and movement of the gopher can be observed, although it is limited by the spatial extent of the study site. Overall, the conclusion I've come to is that the specialized trap is very new in research, and the combination of both methods would allow for a novel method to accurately estimate pocket gophers and other fossorial animal populations. Previously, as far as what I saw in other literature methods for population estimation, it involved rougher counts, and it used capture components that involve killing. The combination of both these methods for research would allow for the animal to not only survive, but also open the door to possibilities such as population estimation across multiple seasons and habitats. Special thanks to Dr. McCleary and our awesome TAs, Catherine and Alex, for a great semester. And I want to thank our viewers for watching, and I'm excited to answer your questions below.